All right, what's up, Richland? So, welcome to our online lecture this semester. We're going to finish Meiosis, which we started on Monday, and then I'm hopefully going to get through the second part of chapter 10, which is really just the last section, 10.6, about how cells control the cell cycle. So let's get into it. Let's keep this as short and brief as possible. So I mentioned already that meiosis is fundamentally different from mitosis in two ways. That it reduces the genetic material of the cell. So you begin with a diploid cell, two sets of encyclopedias, right? Two sets of 23 chromosomes, and you produce four haploid cells, cells that only have one set of encyclopedias. Very different from mitosis. You start with one diploid cell and you end up with two diploid cells that are genetically identical. Here, you're straight up producing four cells that have different number of chromosomes than the cell you started with. You start with a diploid, make four haploid cells. And this is because you go through two rounds of division, meiosis 1 and meiosis 2. And then the second main way is that meiosis generates genetic diversity. So again, mitosis, two identical cells is what you make. In meiosis, you generate four cells that are not only different in chromosome number than the parent cell, but also are genetically different from each other. And that's huge. That is genetic diversity, which is key for evolution by natural selection. And it does this using two mechanisms, which I mentioned already, and we'll get into in this lecture: crossing over and independent assortment. All right, let's go real quick through the first point, that meiosis generates haploid gametes, that it reduces the genetic material of the cell. So I told you it does this by undergoing two cell divisions. So you have two rounds of division in meiosis. You have meiosis 1 and meiosis 2. So here you have a cell which is diploid. So we're going to use a convention that is used by most biology textbooks. And I think it is pretty useful. So in this case, 2N means you have two sets of chromosomes. N is a set of chromosome. And that is a diploid organism, in this case, a diploid cell, right? For humans, N equals 23. So 2 times N, 2 times 23, 46 chromosomes total. You go through S phase, where you replicate the DNA, and now you have 2 times 2N. You've duplicated all of the DNA in the cell, right? So you go from 46 to 92 chromosomes. Now in meiosis 1, you separate replicated chromosome pairs. Remember that the homologous pairs pair up in meiosis, and then, or in prophase, they align paired up in metaphase 1, and then in anaphase, the homologous chromosome pairs are separated. And this actually reduces ploidy. So you go from 2 times 2N to 2 times N, which does not mean you have two sets of chromosome. Still means you have one set of chromosomes, but they're replicated, they're doubled. So it's 2 times N. So if you look at that right under where it says 2 times N, you have, instead of two big chromosomes, a blue and a red one, you only have one big chromosome, replicated chromosome on the top cell, a blue replicated chromosome, one of those blue X's. And in the bottom cell, instead of having two big chromosomes, you only have one of them that is red. And same with the small chromosome, right? So you still have 46 chromosomes technically, but it's only because, but it's still haploid because you only have 2 times N instead of 2N. If you have questions about this, feel free to ask me later. So meiosis 1 reduces ploidy from 2N to N. Meiosis 2 separates sister chromatids, which reduces the chromosome number. So you go from 2 times N to N. You go from 46 chromosomes to 23. And so that's how you end up with 23 chromosomes. The main thing I want you to get here is that meiosis reduces the genetic material in a cell by going through 2 rounds of division. So not only does meiosis reduce the number of chromosomes from 2 sets of 23 to 1 set of 23, but it also generates genetic diversity using crossing over and independent assortment. So let's look at crossing over first. As we've touched on many times already, during replication, homologous chromosomes become replicated homologous chromosomes. So here in this figure, you have a pair of chromosomes, homologous chromosomes. So imagine this is chromosome 1, and blue is the chromosome 1 you got from your father, red is the chromosome 1 you got from your mother. They are both chromosome 1, but they're slightly different, right? Because your mom and dad are slightly different. So these are similar, so we call them homologous chromosomes. Now, during S phase, they're replicated, and they become replicated homologous chromosomes. They become these little X's. And each replicated homologous chromosome consists of 2 sister chromatids that are identical, right? So the X consists of 2 identical sister chromatids, because one sister chromatid came from replicating, doing DNA replication of the other sister chromatid. And these sister chromatids are joined at a region called the centromere, in the center. Think of centromere as the center of the chromosome. Okay, so what I want to point out here is, notice that the homologous chromosomes carry the same set of genes. In this case, we're only highlighting 3 of them, gene A, gene B, and gene D. But homologous chromosomes can carry different alleles. In this case, gene B, the chromosome, replicated homologous chromosome from the mom in red, has big B, capital B, whereas the replicated chromosome from the father, the blue one, has lowercase b. So these chromosomes, homologous chromosomes, carry the same set of genes but different alleles. So they're slightly different from one another. So crossing over happens between the homologous chromosomes, these replicated homologous chromosomes, so that they can exchange alleles. So crossing over is genetic recombination between paired up homologous chromosomes. Genetic recombination is just the exchange of genetic material between chromosomes. And alleles, what ends up happening is that alleles of genes that were formerly on separate homologs can now be found on the same homolog. And when I say homolog here, I mean homologous chromosome. And crossing over happens during prophase 1 when these homologous chromosomes pair up, or these homologs pair up. And the way they do this is there are these proteins, these cohesin proteins and other proteins that form the synaptonemal complex. And the synaptonemal complex is just, think of it as this temporary glue that pairs these homologous chromosomes up. And so once they're paired up, they're going to be so close and they're going to have such similar genetic material, such similar DNA sequence, that crossing over is going to happen. And this is kind of what it looks like. So you have chromosomes that pair up, and as they pair up, they're going to cross over, and the crossing over will happen in between prophase and metaphase. And then in anaphase, when you separate the chromosomes, you end up with chromosomes that now are recombinant chromosomes, chromosomes that have a little bit of the other homologous chromosome, right? A little bit of red and a little bit of blue. And the chiasmata is the site of crossover. It's the little X there where the genetic material is being exchanged, where the chunks of chromosome are being exchanged. And so that contact is maintained until anaphase 1 when they're ripped apart. And then you essentially now have new chromosomes that are a hybrid of the 2 homologous chromosomes. So let's look at one more example of this so you guys I like to show things with different figures from different textbooks so you can get a better idea of what's happening. So here, these two replicated homologous chromosomes are going to pair up in prophase 1 of meiosis. And when they're paired up, they're going to be so close that they're going to exchange some genetic material, right? So there's going to be crossing over that occurs. So after meiosis 1, once you actually have finished the whole first round of division, you end up with chromosomes that are now a mix. You've made new chromosomes that never existed before. Now these chromosomes are a mix of the chromosome 1 you got from dad and the chromosome 2 you got from mom, a mix of red and blue. And these are called recombinant chromosomes, chromosomes that are a mix of the copy that you inherited from mom and the copy that you inherited from dad. And in the end, this is going to result in gametes that carry new recombinant chromosomes, aka genetic diversity, right? So this cell here has only one chromosome for whatever chromosome we're looking at. Imagine this is chromosome 1. But this one chromosome is a mix of the chromosome from mom and the chromosome that you got from your dad. Because remember, this is happening in one individual. This is meiosis. This is happening in the sex cells of an individual. And in those sex cells, meiosis and crossing over are shuffling the chromosome you got from your dad, and the chromosome you got from your mom. All right, so crossing over, one way in which meiosis generates genetic diversity. The other way is called independent assortment. And this is a really simple, sometimes so simple it can get a little confusing principle. But essentially during metaphase 1, remember that replicated homologous chromosomes have paired up and that they line up in 2, right? So they're doing a 2-step. So they line up in 2s, each one with their pair. And the thing is that when they line up, they don't have to be in a certain order. There's not one arrangement that they have to take. So for example, if you look at the first cell here, it's not that all of the blue ones have to be on the left and all of the red ones have to be on the right. It's just random that they ended up that way. So each pair can line up in a different arrangement if it wants to. So if you look at the second cell, you have chromosome 1 and 2, the blue ones are on the left, whereas the third chromosome, the red one, is on the left. And so essentially what's going to happen is you can just draw a line down the middle of these, and this is an anaphase 1 where you're going to separate, right? And you can tell that after you separate, imagine these two cells are just two different possibilities that can happen of how the chromosome pairs will align in the middle. And you can imagine after anaphase 1, in the first cell, you're going to end up with one cell that has only blue chromosomes, whereas in the second cell, you're going to end up with cells that have blue and red, a mix. And the other cells in here are other possible arrangements of how the chromosomes could line up in the center. All right, so let's look at this in another way, right? Again, I want to show you guys the same thing over and over. So after meiosis, you end up with haploid cells. Each daughter cell has only 23 chromosomes, but because of independent assortment, some of these are going to be copies from mom, and some of them are going to be copies from dad. This results in genetic diversity. Essentially, you're shuffling that you have two sets of encyclopedia, and essentially you're going to create cells that only have one set. So you basically are just shuffling those two sets and then splitting them in half. So each cell can get a mix, an assorted mix of the set of encyclopedias is going to be a mix of the set of encyclopedias you inherited from your mom and the one you inherited from your dad. So if you look at this picture, right here, this first possibility of how chromosomes, how the chromosome pairs line up in metaphase, you're going to create cells that either have all blue or all red chromosomes. Each of these cells only has one set, and that one set is going to be either all blue or one red. Why? Because that's how the chromosome pairs arrange themselves in meiosis. Totally random. It just so happened that all the blue ones were on top and all the red ones were on the bottom. But now look at alternative 2. In alternative 2, you end up with haploid cells that do not just have all blue or all red. It's a mix of red and blue. And this is because if you look at metaphase 1 in this possibility, you have 2 blues on top along with 1 red on top. And so whenever you finish meiosis, the 2 rounds of division, you're going to end up with some blue and some red. Same with these other 2 alternatives. You end up with mixes, different possibilities, right, of how you can shuffle the deck, if you will. Okay, so in this example, we've only been considering 3 chromosomes. We have 23 chromosomes, right? 2 sets of 23. These guys here only have 2 sets of 3. So because of that, this is all probability. So if you remember math class, you probably remember taking probability, and that is just, probability is, I hate it. I hate probability, so I hate that I'm even having to teach this, but this is going to drive an important point across. So basically, if you have 2 sets of 3, that can give you 8 different combinations, because that's 2 to the power of 3. 2 times 2 times 2. That's 8. 2 times 2 is 4. 4 times 2 is 8. You have 8 possible gametes. So if you count the different possible gametes as far as their chromosome makeup, you can count them in the example we looked at below. There's 8 different ones. You either have a cell that has all 3 chromosomes blue, all 3 chromosomes red, or different mixes of red and blue, right? Well, you have 2 sets of 23. That's 2 times 2 to the power of 23. That's 8 million combinations, just considering independent assortment. Just talking about how these chromosome pairs can line up during metaphase 1. That is mind-blowing. That means literally guys and girls, we can make 8 million different sex cells, and no 2 will be alike. Not only that, but if you consider that when a guy and a girl get together, the guy can make 8 million different combinations of sperm, or 8 million different combinations of chromosomes in his sperm. The girl can do the same. So 8 million times 8 million ends up being close to 70 billion different possibilities. So literally what that means is a guy and a girl could have 70 billion different children, and no 2 would look alike. And this is only considering independent assortment. This is not to mention that in meiosis, you also do crossing over. And crossing over can shuffle homologous chromosomes. So you're not just inherited blue or red. Each chromosome is a mix of blue and red. So again, the point here is that meiosis generates genetic diversity. So all of the sperm that any guy is listening here is going to be genetically different from each other in your whole lifetime. And same with girls. Girls, you already have generated all of the eggs that you're ever going to release during ovulation, and all of those eggs are genetically different from each other that you will ever produce, right? And again, that's why you will never, ever find anyone exactly like you. Crazy. Genetic diversity. Meiosis generates genetic diversity. let's look at an example of one error that can happen in meiosis, and just how it's so important. It's amazing that when we generate sex cells, that this process happens accurately millions of times, right? Every time we generate a sex cell, meiosis occurs, and it's occurred properly. And if it doesn't occur properly, that's when errors occur that can lead to serious genetic defects. An example of one of these errors is non-disjunction. And this is when chromosomes fail to move to opposite poles during either meiotic division, either meiosis 1 or meiosis 2. Essentially an error during anaphase when it separates the chromosomes. And what non-disjunction literally means is that chromosomes don't, that's the non part, separate, which means disjunction. Disjunction simply means to separate. Non-disjunction means chromosomes do not separate. And so this is an error in anaphase, right? And errors like this can result in aneuploid gametes, which are gametes that are missing or have extra chromosomes. And there's several examples which we've mentioned in class already. Down syndrome is possibly the most common example, and this is trisomy 21, when you have 3 copies of chromosome 21. This is because of non-disjunction. During meiosis, one of the gametes, either the father or the mother, did not separate chromosome 21. So you end up with 2 chromosome 21s in that gamete. And then when that gamete fertilizes the other gamete, when the sperm, say for example, that has 2 chromosome 21s, fertilizes the egg that has 1 chromosome 21, you end up with 3. Klinefelters, similar. You have 3 sex chromosomes, 2 X's and a Y. You have an extra X chromosome. Turner syndrome, only 1 X. Klinefelters, by the way, is going to be a male, technically, because it has a Y. Turner's is going to be a female, even though there's only 1 X chromosome. And these errors in meiosis, like non-disjunction, are the most common cause of spontaneous abortions in human. And again, this is because the developing embryo has a checkpoint set in place to tell whether it has the right number of chromosomes or not. And if it doesn't have the right number of chromosomes, there's a mechanism set in place to self-abort, basically, to protect the developing embryo from serious genetic disorder. Okay, so let's look at how this happens. Again, non-disjunction. Chromosomes don't separate during anaphase 1 or anaphase 2. In this example, this is non-disjunction in meiosis 1, in anaphase 1. So after meiosis 1, you fail to separate the replicated homologous chromosomes, and they go to 1 cell. Imagine this is only chromosome 21, for example. And then after meiosis 2, you end up with abnormal gametes that have a complete set of chromosomes plus 1 extra one, in this case an extra chromosome 21, and the 2 on the top. Whereas the 2 on the bottom are also abnormal because they have a full set missing 1 chromosome. The ones on the bottom don't even have chromosome 21. Now imagine though that a sperm fertilizes the top 2 eggs. That sperm is going to carry 1 copy of chromosome 21, as it normally should. It's only going to have 1 set of encyclopedias, right? And after it fertilizes them, you're going to end up with zygotes, which is a single-celled state of an organism, of a multicellular organism. Zygotes that have 3 copies of chromosome 21 instead of 2. And in the bottom, you actually are going to end up with zygotes that only have 1 copy of chromosome 21, which those never live, right? Whereas, obviously, we probably know or have seen someone with Down syndrome, so we know that at least the organism can tolerate, the human body can tolerate 3 copies of chromosome 21. Now, this can also happen if there's a failure in anaphase 2. So here in meiosis 2, we'll look at meiosis 1 in this example. Anaphase 1 occurred properly. The chromosome pair separated. Now, here, FYI, by the way, here the homologous chromosomes are in the same color, but you should know that one of those replicated homologous chromosomes is from this individual's mother, and the other one is from this individual's father, right? It's the homologous pair this individual has. So meiosis 1 occurs normally, the homologous chromosomes separate, but then meiosis 2, where you separate the sister chromatids, the cell on the bottom undergoes non-disjunction. The chromosomes don't separate. So you end up with one abnormal cell that has a full set of chromosomes plus 1, n plus 1, and the plus 1 here would be chromosome 21. So it has two chromosome 21s, and the cell on the bottom has a full set of chromosomes minus 1. It has 22 chromosomes because it's completely missing chromosome 21. So then imagine that a sperm comes along that has one set of chromosomes. It fertilizes the embryos, the eggs on the bottom. You're going to end up with one cell, or if it fertilizes the egg that has two chromosome 21s, it's going to produce a zygote, which will become a human that has 3 copies of chromosome 21, Down syndrome. And the other one is only going to have 1 copy of chromosome 21. It's never even going to make it, right? It's probably going to spontaneously abort. And so I want you guys to know this error, non-disjunction, and understand at what step of meiosis it occurs and what it consists of, what causes, what happens during non-disjunction. Okay, and that's it as far as meiosis goes. And so here's a summary of meiosis and mitosis side by side, which I think is really helpful. So the main differences are in rounds of division. Mitosis goes through 1 round, meiosis goes through 2 rounds, which is why you end up with haploid cells. You reduce the number of chromosomes by half because you go through 1 extra round of division. Crossing over does not happen in mitosis. It does happen in meiosis. All right, and the crossing over happens in prophase 1. Metaphase alignment. In mitosis, you have a line dance, a single file, the homologous chromosomes lined up individually. In meiosis, you have a 2-step, the homologous chromosomes pair up, right? They've already paired up in prophase 1, and in metaphase 1, they're going to line up in pairs. They're going to 2-step. Then the end result, mitosis, 2 diploid cells, meiosis, 4 haploid cells. And then genetic diversity in meiosis, only in meiosis, is achieved by crossing over, independent assortment. Crossing over shuffles the alleles of each homologous chromosomes, or I guess here I should say shuffles the alleles between homologous chromosomes, and it happens during prophase 1. Independent assortment is the other way that genetic diversity is achieved by meiosis. And this is where homologous chromosomes lined up independently of other chromosomes during metaphase 1. This is independent assortment is also called random alignment. You'll see that in some textbooks as well. But this essentially shuffles the homologous chromosomes each daughter cell can receive. So each cell is going to receive a shuffled set of encyclopedias, if that makes sense. All right, and that's it. Feel free to take a break now, pause this, go stretch, exercise, play a video game, grab some coffee with a friend, get a nap in, and then come back and watch this last part of the lecture. Okay, so this last part is the last section of chapter 10, 10.6, and it really has to do with how cells control cell division. So I wanted to do this after meiosis because it's easier to just compare mitosis and meiosis and the steps and what makes them different, and then come back to how does a cell even control cell division? How does it control the cell cycle? So some of this stuff is going to be similar for meiosis and mitosis. But just kind of a review slide of the cell cycle. So remember, the cell cycle consists of 2 main steps, interphase and M phase. Interphase is a phase that is in between each division, hence interphase. Consists of gap phase 1, synthesis step, and gap phase 2. And then you have M phase, which consists of mitosis and cytokinesis. This is a crazy cycle. And in order for it to happen properly, you have to be able to control it, right? And actually, we'll look at this in a little bit. Whenever the cell cycle is not controlled properly, that is when cancers arise. So, how does a cell control the cell cycle? This observation initially came from these kind of experiments where you would take the cytoplasm, you would straight up take a needle and puncture a cell that's undergoing division, undergoing mitosis, remove the cytoplasm. I guess this is an oocyte, so this oocyte is undergoing meiosis. So you would straight up take some cytoplasm, inject it into an oocyte that is not dividing, it's not undergoing meiosis, and all of a sudden this oocyte starts meiosis one, time and time again. Let me repeat that. You remove some cytoplasm from an oocyte that is undergoing meiosis, inject it into an oocyte that is arrested or that is not dividing, and now this oocyte is going to start dividing. Here's another example. You take a cell that is undergoing mitosis, an M phase, and you fuse it together. Sometimes this happens if you put two cells very close together and destabilize their membranes, they can fuse together. And right after fusion, the cell that was not dividing is going to start dividing. So how would you interpret the experimental results shown here? I'll let you think about it while I eat some grapes. So the most straightforward interpretation here was that there are some factors in the cell during mitosis that trigger cell division, that stimulate cell division. So you can take cytoplasm from a cell that's dividing and you take some of these factors that promote cell division, inject it into a cell that's not dividing, and these factors will now actually tell this other cell to start dividing. So again, there must be factors that control the initiation and progress, actually, of mitosis. And since then, scientists have discovered a complex cascade of enzymes that are involved in initiating and driving mitosis. And these factors were discovered using frogs, sea urchins, clams, yeast, and fruit flies as model organisms. Remember, scientists, they can't always do experiments on human cells or on humans, right? So we use model organisms. And believe it or not, sea urchins and clams are model organisms. And so the main driver of cell cycle progression is a complex of two proteins. CDKs, which stands for cyclin-dependent kinases, and these are enzymes that phosphorylate other proteins. And cyclins, which activate CDKs. Actually, cyclins are the C in CDK. CDKs are cyclin-dependent kinases. And so here, right below that, is a figure of these two proteins complexing together. So you have a CDK and a cyclin that are interacting. And what you'll notice in the graph to the right, it's talking about the concentration of cyclins. And on the bottom is the progress of the cell cycle. So the different steps of the cell cycle, G1, S, G2, and M phase. And so you'll notice that in red are the levels of cyclin. And the levels of cyclin peak during mitosis and then dramatically drop after mitosis. And as soon as they start increasing, the cell enters M phase and then they drop. And as the levels of cyclin raise and drop, the activity of CDKs, which are enzymes, they're enzymes that phosphorylate other proteins. The activity of CDKs turn on at the peak of cyclin levels, right? So you see that as the level of cyclin, the concentration of cyclins peak, the red bar, it peaks, then the CDK activity turns on. And right after cyclins, the level of cyclins drop, CDK activity turns off. So CDKs, which are the enzymes that are actually driving the cell cycle, turn on and off by cyclins. So that's why they are cyclin-dependent kinases. So as the figures show, cyclins come and go at regular intervals. In the top figure, we're looking at just cyclins in general. On the bottom, you can see that there are different cyclins. In the bottom graph, you'll see that there's different color lines for the different cyclins. Cyclin D, cyclin E, cyclin A, cyclin B. They control the different steps of the cell cycle. So you'll see that if you're in G1 phase, you will transition to S phase at the peak of cyclin E expression. When cyclin E levels are at their max, that's when you transition to S phase. And then when cyclin A levels are at their max, you transition to G2 phase. And then when cyclin B levels are at their max, you transition into M phase, into mitosis. What this graph, neither of these graphs show you, is that CDK levels stay constant. Remember, CDKs are the cyclin-dependent kinases. These are the enzymes that are actually, you can consider them the go signal for cell division. And it's their activity that turns on and off depending on the presence or absence of cyclins. So as soon as cyclin E starts getting made and the levels increase, they're going to turn on a CDK. And then as soon as cyclin E drops, that specific CDK is going to turn off. And then cyclin A is going to increase, and that's going to turn on its own CDK, which is going to drive the cell from S phase to G2. And as soon as that cyclin decreases, that CDK turns off. But the level of the CDKs are not changing. They're constant. They're just being turned on and off by the cyclins. That's why they're called cyclin-dependent kinases. And like I said, each phase of the cell cycle is characterized by its own cyclins. Okay, now this cell cycle has checkpoints that ensure that everything is looking good before the cell moves on to the next phase. And so these checkpoints, think of them as airport security checkpoints, where you have these TSA security guards that pat you down and make sure you're good to move on to the next step, right? From outside of the airport to inside of the airport. Here you have checkpoints that make sure you can go from the G1 phase to the S phase, and then from the G2 to the M phase, and then from metaphase to anaphase. And so you have these different checkpoints. And in this first checkpoint, the G1 to S phase checkpoint, basically the security guards are asking, is the cell ready to begin cell division? Does it have everything it needs in order to begin DNA replication? And once this is passed, the cell commits to division. Then it starts replicating DNA, and once you replicate DNA, you're committed. You will divide, right? The next checkpoint is a G2 to M phase transition. And here, the cell is asking, did the DNA replicate without error? And if so, then it can move on to M phase, to mitosis and cytokinesis. And once this checkpoint is passed, the cell commits to mitosis. And then there's a late metaphase or spindle checkpoint, which asks, are all chromosomes attached to spindles and are they lined up properly? If this is not the case, what happens in any of these checkpoints, if the answer is no, is the cell cycle is going to stop right there, and it's not going to go on until the errors are fixed. And if those errors are not fixed, then the cell will not go on. The cell cycle will not go on until it's fixed. And if it's never fixed, then that cell will die. It will actually kill itself in using a program called apoptosis, or programmed cell death. So at metaphase, If all the chromosomes are not lined up in the middle, the cell will not proceed to anaphase. But once all the chromosomes are lined up in the center in the metaphase plate, then the checkpoint is passed, and the cell will proceed to go through anaphase, telophase, and cytokinesis. And here's a little visual reminder. Think of these checkpoints as the TSA airport security of the cell. So these checkpoints have stop and go signals. The go signals are these CDKs and the cyclins that we talked about. They basically tell us, okay, go through the checkpoint. And then it also has stop signals, green light, red light, right? The red light are these proteins called tumor suppressors. And they're proteins that will stop the cell cycle if it hasn't met the standards of that security checkpoint. So some of the go signals, you don't have to know these, but just so you know, we've identified some of these CDK cyclin pairs. So for the G1 phase to S phase transition, we have CDK2 and cyclin E. For the G2 to M phase transition, we have CDK1 and cyclin B. And for that late metaphase checkpoint, we actually have another protein called, or proteins called the anaphase promoting complex. So these are all go signals that say, go to the next step, go to the next step. But if things are not right, then you have these stop signals. And the tumor suppressors, think of them as basically if you go through the metal detector at the airport and the metal detector goes off, the security guard is going to tell you to stop, and it's going to check you. And it's going to try and fix that mistake, or it's going to tell you to fix that mistake. Like, hey, take out any metal things from your pocket. If you go through that checkpoint again and it still goes off, that security guard might get more suspicious. Eventually, he might pull a gun and shoot you if you don't drop your weapons or whatever it is you have, right? That's kind of what tumor suppressors do, is they will stop the cell from going on to the next step of the cell cycle until it fixes the problem, and if it doesn't, it kills the cell. So going back to the go signals, to the CDK and cyclins, what controls those cyclins? We said that the cyclins control the CDKs. They turn the CDKs on. So what controls the cyclins? And one of the many different ways this can happen is by growth factors. Growth factors are proteins or hormones released by other cells that will tell the cell when to divide. This is especially true during development, human development. You have hormones and proteins that are being released, these growth factors, that are telling the cells when to divide. So here in this figure, you have a growth factor that bind to its receptor. And a receptor is a protein that spans the membrane. It's a transmembrane protein that binds to that growth factor. And when it binds to that growth factor, it begins a cascade of kinases, these proteins that phosphorylate other proteins, that will ultimately activate cyclins. So if you follow this cascade of kinases, RAS activates RAF, which activates MEK, which activates ERK, right? I'm at step 4 of this. And then ERK in step 5, this ERK is going to move, this protein is going to move into the nucleus, and it's going to activate RB and E2F. E2F is a transcription factor, which is going to bind to the cyclin genes and activate transcription to make mRNA, and then the mRNA is going to be translated into protein, into cyclin protein. And then those cyclins are going to turn on the CDKs. But that is essentially how you control cyclin levels. And then cyclins can turn on CDKs. Okay, so let's talk about cancer. What the heck is cancer? It has to do with the cell cycle. Typically, cancers arise from mutations that affect cell cycle control genes. So these stop and go signals that control the cell cycle, when they're affected by mutations, the cell begins to divide precociously, or begins to divide in an uncontrolled manner, which can result in a tumor. And remember, mutations are changes in the DNA sequence of a gene, and they're typically errors made during DNA replication before the cell divides. Or they can happen when you come in contact with an agent or a chemical that causes mutations. These are known as carcinogens. So, for example, all of you cigarette smokers out there, cigarette smoke contains many carcinogens, many chemicals that when they contact your lung, they mutate DNA in your lung cells, and then some of those mutations can hit cell cycle genes, which will end up causing lung tumors. So mutations in cell cycle genes can do two things. They can hyperactivate genes that stimulate cell division, the go signals, which is basically like pressing on the gas of the cell cycle, telling it to go faster. And they can inactivate genes that stop cell division. They can basically inactivate the brakes, the stop signals, right? These tumor suppressors. So then imagine now, not only are you pressing on the gas, but you have no brakes to stop the car if the cell cycle is the car, right? Progressing. And so now you basically have uncontrolled cell division. So let's talk a little bit more about these two kinds of genes. So you have proto-oncogenes, which are these go signals, which are the gas pedal of the cell cycle, right? They're normally involved in cell cycle progression. These are like the CDKs and the cyclins that tell the cell when to divide. So they're good. They're good genes, these proto-oncogenes. And proto-oncogene, proto simply means before, and oncogene means a gene that promotes cancer. Onco means cancer, gene is gene. So it's like cancer genes. So normally these are good genes. Proto-oncogenes are good genes. And when they're mutated, they become oncogenes, right? So there you can see normally they're proto-oncogenes. When they get mutated, they become oncogenes, and then essentially you're pressing on the gas pedal. They're the gas pedal of the cell cycle. So you're telling the cell to divide even when it shouldn't. And then the other kind of genes are the stop genes, the stop signals. So these are the tumor suppressor genes. And they're normally involved in the cell cycle checkpoints. They're the TSA security guards, right? The ones that will shoot you if you do not empty out your pockets, or if you do not drop the weapons that you're carrying. So these are the brakes of the cell. So cancer cells, they not only have to activate oncogenes, they have to inactivate tumor suppressor genes. So they have to cut the brakes so that the cell cycle can accelerate and not have to brake and not have to stop. So these are the two kinds of genes that have to be mutated in order to result in cancer. So the inactivation of tumor suppressors and the activation of oncogenes causes cells to divide in an uncontrolled manner and form a tumor. So here in this figure you have normal cell division. These cells are dividing normally. Again, they're going to divide, except in this case, that one cell is going to divide into a normal cell and into a potentially cancerous cell, a cell that has randomly acquired a mutation, right? Created a mutation in either a tumor suppressor or a proto-oncogene. And this cell, once it accumulates more mutations, so that now you're pressing on the gas and you've cut the brakes, no more brakes, then it begins to form a tumor. And this tumor can be benign if it doesn't affect surrounding tissues. So just because you have a tumor doesn't mean you have cancer. You can have a tumor, get it checked out, and it can turn out to be benign, and you're good. You're not in danger of dying. But if that tumor is malignant, then it becomes a cancer. Then this is what we know as cancer. And a tumor is malignant when it invades, or if it invades, and affects surrounding tissues. And malignant tumors can spread or metastasize. So this happens when cells acquire the ability to break away from the original tumor. So normally cells don't just randomly um, break away from tissue, right? Your skin cells don't just randomly break away from your skin. They stay together. That way that's how your skin stays together and it doesn't just fall apart. But these malignant cancer cells can acquire the ability to break away from the tumor and start new tumors in other tissues or organs. And so this is what we call metastasis. Whenever you hear someone say, "Man, their cancer is spreading," it's that the cells are breaking off of the tumor and spreading to other parts of the body. And so ultimately, people that die from cancer don't die because bacteria are attacking them or viruses are attacking them. They die because the tumor, the original tumor, is spreading to other organs, vital organs. And so your lungs can stop working if you have lung tumors. And your brain can get a tumor. And your kidney, your liver can get tumors. And then obviously, if those organs are affected to where they can't work, you will eventually die because of that. So cancers kill you because they spread to vital organs. All right, so let's I want to give you an example of a tumor suppressor. We already looked at an example of a proto-oncogene, which are the cyclins and the CDKs. So those can be mutated to where they're constantly active and constantly telling the cell to divide. I want to give you an example of a tumor suppressor, which are the TSA security guards. And P53 is a gene that makes this protein that can kill the cell if there are errors that are not being repaired during the cell cycle. And it is a tumor suppressor poster child, which simply means a classic example of a tumor suppressor. It encodes a protein that plays a key role in the G1 to S phase checkpoint. And what this protein does is it monitors the integrity of DNA. Basically, make sure that there are no mutations or breaks in the DNA. If the DNA is damaged, P53 will halt cell division and activate repair enzymes to repair the DNA. So this is what you can see here in this figure. There's a DNA break. P53 is going to sense that, turn on DNA repair enzymes, activate those so that they can repair the DNA breaks. Now, if the DNA break is repaired, P53 is going to allow the cells to continue with cell division. Basically, the security guard is going to be like, okay, you can go to the next step. If it's not repaired, if the DNA damage is irreparable, it activates a self-destruct program where it literally kills the cell. Right? So I wasn't joking about that. These tumor suppressors will literally shoot you. And why? Because this minimizes the possibility of passing on mutations to other cells. Now, you can imagine that when P53 is mutated or inactivated, right, the damaged cells can, cells that have mutations in their DNA can divide and form tumors with no problem. Or I guess I should say cells can divide even if they have mutations, and eventually those mutations are going to become, are going to affect other genes like proto-oncogenes. They're going to activate proto-oncogenes into oncogenes and then eventually cause that cell to become cancerous and form a tumor. And this, it's interesting because if you look at all of cancers, over 50% of these cancers have mutations in P53. And proto-oncogenes, like I mentioned, they're normal cellular genes. They're necessary. They're the go signals. And they become oncogenes when they're mutated. So they have to, proto-oncogenes are not bad unless they accumulate a mutation that turns them into an oncogene. And these oncogenes are going to drive cancer by promoting cell division. when it shouldn't occur. They're going to be telling the cell to go on to the next step, even if it shouldn't go on to the next step. Some of these proto-oncogenes encode receptors for growth factors. So basically, if the receptor is mutated, it stays on, and it's telling the cell to divide over and over and over again. Some of the proto-oncogenes encode signaling proteins that promote cell division. Some of these are kinases like RAS and SRC. Some of these are transcription factors like E2F. Some of these are CDKs and cyclins, right? Yeah. Now, final slide, and then I'll let you guys go. A few things about risk factors, because this affects you directly. There's a lot of lifestyle factors, specifically what you eat. What you eat, believe it or not, has been associated with cancer risks. Doesn't mean that eating bad food causes cancer, but it does mean that if you eat bad food, you're more likely to get cancer. And this is typically high-fat, low-fiber diets, aka fast food diets. So these fast food diets have a ton of animal fat, which is not great for you, and it has very low fiber. And this is shown to be correlated with increased risk in colon cancer. Lack of exercise has also been shown as a increased risk factor of cancer. So don't just get on these machines that have bands that jiggle your butt. Actually get out there and do some exercise. Go running, lift some weights, do some yoga. Anything is better than sitting on the couch. Now, exposure to carcinogens will also increase your risk of getting cancer. Smoking has been shown to be a key cause of lung cancer. Exposure to UV rays causes melanomas or skin cancers. So being out in the sun without sunscreen will, there's a high risk of getting skin cancer. Going to tanning beds these tanning beds are UV rays that cause mutations in DNA. They're also gonna give you a greater risk of having skin cancer. So wear sunscreen. If you inherit certain mutations, you're at a greater risk of having cancer as well. So there is a genetic component to cancer. You can pass on, or you can inherit mutations from family members that will predispose you to cancer. So there's mutations that can predispose you to breast cancer, to colon cancer, to lung cancer, many mutations that affect some of these cell cycle control genes can basically put you at odds where now you're more likely to get cancer. And then the last thing that none of us can avoid is aging. Aging is the ultimate risk factor for cancer. If you live long enough, you will get cancer. That is virtually guaranteed. There are exceptions of people that live over 100 that don't get cancer, but that is a very small minority. The majority of people will get cancer sooner or later. So even if you eat incredibly healthy, never are exposed to carcinogens, don't inherit any bad mutations, ultimately aging will get you. But the goal here is to live as long and a full life as possible. So I encourage you guys, go out, live a long and full life. If you're smoking, stop smoking. If you're eating McDonald's, start eating healthy. If you're not exercising and just playing video games, go out and get some exercise. If you're not wearing sunscreen, wear some sunscreen when you go outside. And enjoy life. All right, guys. See you on Friday.